welcome. Thank you so much for coming today, to those in person and on Zoom. On behalf of the Invert Ecology Lab, we've hosted. Thanks for the lab for putting together the spread. And we're excited to welcome today's seminar speaker, Dr. Paul Bourdeau. He grew up in southeastern Massachusetts. He got his BS biology and his master's of marine biology at the University of Massachusetts in Dartmouth, and then went on to do a PhD in ecology and evolution from Stony Brook University in New York, working with Diane Padilla. He then did a postdoc at Michigan State University, when he then joined Cal Poly Humboldt in 2014, and there he wears many hats. So he is an associate professor of marine biology and ecology. He's also the graduate coordinator for the Department of Biological Sciences and the interim director for the Telonaker Marine Laboratory. Did I say it right? Yes. It's practicing in my head. All right, and his lab's research focuses on how marine organisms respond to environmental changes, particularly those induced by human activities, such as the introduction of non-native species and climate change. So please join me in welcoming Dr. Bourdeau for his talk, The Effects of Environmental Change on the Importance of Non-Consumptive Predator Effects in the Rocky Intertidal Zone.
Okay, can everybody hear me okay? In the back too? All right, well, first of all, thanks so much for the invitation. Sarah, thanks for the invitation. Amanda, thank you for hosting and for that introduction. I've spent a fair amount of time at a handful of the iconic marine labs on the West Coast, but I've never been here before. And so I'm really excited to get this invitation and also share my work with you. So let's start talking about that. So as Amanda mentioned, our lab, my students and I kind of focus on broadly how does environmental change influence species interactions, which is a pretty broad umbrella. And so if we were to have sort of an undercurrent theme or a theme that runs through that is looking at how environmental change affects predator-prey interactions. And so I just want to outline some of the sort of questions we'll be addressing today in the talk. We'll start out talking about how the loss of predator abundance and diversity affects prey behavior, and then whether or not prey can respond to field-relevant risk cue concentrations, and I'll explain what that means a bit later. And then, does natural variation in predator abundance influence the importance of these non-consumptive effects, which we'll define shortly? And then, how do environmental factors limit predator risk cue persistence and prey's ability to respond to those important cues? So why do we study predator-prey interactions? Well, as most of you probably know, right, predation is a major structuring force in ecological communities, right? And since the dawn of modern ecological research, right, investigators have been examining and documenting the importance of predators and regulating the populations of their prey. And of course, the effects of predation don't just affect prey populations, but they cascade onto other members of the community, right, namely the prey's resources and other community members that might be using those resources as well. And I probably don't need to go through this example to this group here of the classic trophic cascade involving sea otters consuming sea urchin grazers, reducing the urchin population density, which then results in a release for forest forming or canopy forming and understory kelp, right? So cascading effects of the predator onto the entire community. But predators can really have two types of effects on their prey, right? The aforementioned consumptive effects, where predators are actually consuming prey, leading to their death and reductions in density, but also non-consumptive effects, right? Where the mere presence of a predator is actually going to influence the behavior of the prey who's trying to avoid being eaten, right? But that avoidance, even though it increases survivorship of the prey, has fitness trade-offs associated with growth and reproduction. So if we just look at this example here, you imagine in an environment where predators are rare and there's very little risk in the environment, prey species can go about their business of feeding and eating with little vigilance, high activity levels, can consume a lot of food, and as a result can produce a lot of offspring, right? If you enter predators into the equation, however, right, that creates risk in the environment prey are going to spend less time active, less time searching for food, maybe more time hiding. And of course, if there's less time eating and feeding, then you might have fewer energy reserves for reproduction, and you might produce fewer offspring. But of course, so there's a cost associated with the avoidance of the predator risk in the environment. Of course, there's a trade-off, right? If you didn't spend time avoiding being eaten and you get eaten, right, your future fitness immediately drops to 0. Now, these non-consumptive effects don't just affect prey populations either. They can also cascade onto other members of the community through what we call trait-mediated indirect interactions, right, where the predator changes some trait in the prey, in this case behavior, right? The prey is going to reduce its activity, maybe increase its vigilance or hiding. And then there's no reduction to the density or abundance of the prey, but its activity changes, right? And then the predator can have cascading effects onto the prey resources just by altering the behavior of the prey. And I love this example here. So this is an absolutely crazy study that was done in some Pacific Coast islands where they did audio playback of some large carnivores, and those sounds from the larger carnivores scared the smaller carnivores, in this case the raccoons, from foraging in the intertidal. And because the raccoons were spending less time foraging in intertidal in these areas where the large carnivore sounds were being played back, the smaller invertebrates that the raccoon were preying on were released from predation from the raccoon and increased in abundance. One of those, red rock crabs, because the raccoons were gone from the intertidal zone, the red rock crabs could move further up into the intertidal zone because their predator wasn't there to eat them, and that allowed them to outcompete these tide pool fish for a shared prey, in this case a carnivorous whelk that they both liked. So these non-consumptive effects can also have cascading effects on the entire community. And what we know from several decades now of research on these non-consumptive effects is that these non-consumptive predator effects can be just as large or even larger than consumptive effects of predators in many systems. And if you look at meta-analyses comparing the sort of standardized effect size of consumptive effects and non-consumptive effects across lots of ecological systems, these non-consumptive effects can be up to 63% times larger than non-consumptive effects. But theory predicts that these non-consumptive effects and these trait-mediated indirect interactions that result from them should actually strengthen in terms of their magnitude and importance as predator density increases, right? And the reason for that is that prey responses to predators should be risk sensitive, right? The prey response to predators should increase as predator density increases because these prey are balancing these trade-offs, right? So if risk is higher, you're going to enact your anti-predator defenses more often. But even though we have lots of great research on these non-consumptive effects, there's very few studies that have actually manipulated predator density in the field in a realistic way, or have used natural predator density gradients as a way to assess this theoretical prediction about the strength of non-consumptive effects. Not only that, but a lot of what we know about the strength and importance of non-consumptive effects in ecological systems actually come from laboratory-based studies, with a few notable exceptions. And in those lab-based studies, for example, in marine systems, right, one of the major ways that prey species respond to the presence of predators are through waterborne chemical cues, right? And so a lot of these lab experiments looking at the non-consumptive effects of predators on their prey mediated through these chemical cues oftentimes use unrealistic predator densities, which increases or overinflates the amount or concentration of cues in those experiments. Now, I'm not being critical of those experiments. I've done those experiments myself, and we've learned quite a bit about non-consumptive effects through those types of experiments. And it's actually really difficult to evaluate prey behavioral responses to predator cues out in nature, especially over large spatial scales. So we have some knowledge gaps there, because if we're oversaturating risk cues in these laboratory experiments, we might be overestimating the importance of these non-consumptive effects in these systems, right? So what I want to talk about today is kind of what we're doing in my lab to sort of address some of these knowledge gaps about non-consumptive effects. And I really want to focus today on student research projects in the CSU, right? We're not R1 universities, and so a lot of us PIs rely on our students to do research. And so I really want to highlight my students' research. I've been really lucky over the years to get some really motivated, really dedicated, and super curious graduate students and undergraduate students who really sort of launched this particular line of research in my lab. So what we've been doing to sort of address this idea that non-consumptive effects and their importance should increase with increasing predator density is by taking advantage of a natural experiment in nature's laboratory, the intertidal zone, right? And again, I'm probably pretty sure I don't have to explain to you about the sea star wasting outbreak that happened between 2013 and 2015 that decimated lots of intertidal and shallow subtidal sea star populations along the west coast of North America, including the ochre sea star Pisaster ocratius. And even though some of those populations started to rebound and recover fairly rapidly as soon as 2015, in fact, I'm not sure if this works, but what you can see from the map here, so 2015, 2016, 2017, left to right, you can see the dark dots there, dark symbols are populations that are in recovery since the outbreak. The orange dots are not in recovery. And what you can see through time is that there's been really variable recovery. And if I can point you to, I guess I'll walk up here. So this is our neck of the woods up here, up to the Oregon border, down around Cape Mendocino here. Through time, this variable recovery has led to a natural Pisaster gradient of increased recovery in abundances to the north, decreased recovery in abundances to the south. So we're sort of taking advantage of that natural variation in predator density. Oops. Did I mess something up by pressing this maybe? It just stopped moving forward here. Maybe we can do it this way. I can do it that way. Okay, so most of you know Pisaster as the original keystone species, right, because of its predation on foundational mussel species in the intertidal zone. But Pisaster is also a very important predator of black turban snails, Tegula funebralis. In particular, on the northern California coast, where mussel recruitment is relatively low, especially relative to Oregon and Washington, and boulder fields rather than rocky benches are sort of the predominant habitat, barnacles and tegula are the main prey resources for Pisaster. So Pisaster can have consumptive effects on these black turban snails, and in some places can consume about 25 to 30% of the adult tegula population in a year. But like most prey, these turban snails just don't sit around and let themselves be consumed by Pisaster, right? They can actually respond to the physical touch of Pisaster, but they can also respond to the chemical scent of Pisaster as well. So if you make some Pisaster tea, you condition Pisaster in a bucket and then introduce that seawater that's been conditioned with Pisaster to tegula. Probably all of you know from classroom experiments that tegula will crawl out of the water and sometimes all the way out of the tank to avoid being eaten by Pisaster. And this is actually a very common and well-documented response in natural tide pools as well. And you can see it in this photo here. There's the Pisaster at the water line, and you can sort of see the black turban snails just out of Pisaster's reach at the top of the tide pool there. So this makes for a really tractable and convenient study system to look at the effects of predator density on the importance and magnitude of non-consumptive and consumptive effects, and in particular with this natural experiment of wasting loss and variable recovery to look to see how predator density influences non-consumptive effects in the intertidal zone. Just a quick, there's a low battery warning up here on the computer, so I don't know if I should plug it in or... It's possible I did that. Okay, sorry. Okay, so the way that we started actually investigating this larger question started with an undergraduate student of mine at Cal Poly Humboldt, Kendall Murray, who's now a doctoral student at the University of Washington. And what Kendall was really interested in is how might the loss and variable recovery of Piezoster influence non-consumptive effects on tegula within the context of predator diversity in the intertidal. And so she really took the approach of looking to see whether variation in Piezoster abundance, both in terms of density and biomass, but also Piezoster in the context of other potential predators of tegula. In the absence of Piezoster, can these other potential predators, including the red octopus and the spot-bellied rock crab, can they sort of step in and fill the role of Piezoster in terms of inducing these non-consumptive effects on tegula? And so she did this really complex and massive experiment that I urged her not to try, but she pulled it off. And so this just sort of gives you a schematic of all the different experimental treatments that she did. So she varied, as I mentioned, Piezoster abundance, Piezoster biomass, Piezoster size, and then also compared Piezoster to octopus, rock crabs, and then different combinations of those predators to look to see how the chemical cues from these different treatments would influence tegula crawl-out behavior and grazing on macroalgae in the lab. And there's a picture of just what, so that's just one temporal block of the experiment, which had to be done over multiple time periods because of the size of it. That's a good eighth of our entire marine lab there taken up. Okay, so let's just walk you through what she found. So here's our plot here on the x-axis of the treatments, and then these are just the tegula that are crawling out of the water in response to these different chemical cue treatments. And let's just cut to the good part here. So what I want you to focus on here are the highlighted bars, because what you should notice here is that in control seawater, right, seawater that hasn't been treated by any sort of predator water at all, right, the tegula are pretty happy to graze on the algae at the bottom of the aquarium. But when Piezoster are around, whether it's 2 large Piezoster, 3 small, or Piezoster with any other predator cue, right, the tegula are out of the water. But what we thought was really fascinating is that even though there's documented evidence of octopuses preying on tegula in the field, and certainly rock crabs, and Dr. Geller knows about this, preying on tegula, the tegula up in our neck of the woods did not respond to the chemical cues of those predators by crawling out of the water. And obviously, if tegula is spending more time crawling out of the water, they're going to spend less time feeding, right? So this is just the kelp consumption by those tegula. Again, what you'll notice is it's only the Piezoster cue treatments that induced any sort of grazing suppression in tegula. And obviously, if you're spending more time out of the water, less time eating, you're going to grow less as well, right? So here's that non-consumptive effect of these cues on the snails. And again, same story here, where Piezoster is really the only chemical cue that tegula is responding to. So that's great. That's in the lab, and that was pretty interesting. So we wanted to do at least get some bit of pattern data that might support these laboratory results. And remember, this was just a senior thesis here, so we couldn't give Kendall a master's for this. So I tried to limit what she did here, but she went out to these 4 different sites where we had measured Piezoster abundance. And there's our gradient of Piezoster abundance across our 4 sites there. And what she then did was look to see what was the proportion of tegula high in the intertidal zone across these 4 sites. And what we found is there's a pretty significant and positive relationship between the proportion of tegula up in the high zone with Piezoster density here. And interestingly enough, if you look at the relationship between tegula vertical distribution and the other 3 predators that we looked at in the experiment, you don't find any relationship at all. And that also includes the 6-armed sea star, Leptosterius, although we should note that those densities are really, really small. Up in our area, Leptosterius did not recover nearly as quickly as Piezoster did. So we didn't have good natural densities to sort of look at that relationship. So take that upper right box with a grain of salt. But with the others, no clear relationship. So the field patterns sort of support the mechanistic stuff that we found from the lab experiment, suggesting that, Piesaster significantly affects tegula behavior and vertical distribution. But the diversity of predators that we have up on the far northern California coast doesn't really provide functional redundancy in terms of these non-consumptive effects that Piesaster is having on tegula. And so suggests some potential ecosystem instability, particularly in the low intertidal zone, if Piesaster is removed from the system. Okay. Just looking at the time. One of the other really interesting things that came out of Kindle's experiment is that when we look at either Piesaster density or Piesaster biomass using the replicate aquarium that we used in our experiment, we see this density-dependent response, or this risk-sensitive response by tegula to Piesaster cues, right? Supporting what theory suggests, that prey responses to predators should be risk-sensitive in response to increases in predator density. Now, what we did with Kindle's experiment is we used a density of Piesaster that was within reason for what we see in the field, but it was at the high end. And so what we really wanted to know was whether or not tegula could respond to Piesaster chemical cues at field-relevant concentrations, right? Sort of addressing that gap that a lot of studies sort of oversaturate Q in the environment. And so we want to make sure we weren't overestimating this effect of Piesaster. So what we did is we took those aerial densities of Piesaster that we had in the field, and we converted those to volumetric densities, right? To sort of give us a very coarse, but we think reasonable proxy for what the chemical Q density might be for Piesaster in the field. And my master's student at the time, Tim McClure, followed up Kindle's experiment with this elaborate experimental setup of his own, where he again had these header tanks where he conditioned seawater with Piesaster, but the seawater flowing through those conditioning tanks then flowed through a set of cascading cups, which diluted the Q coming from the sea star tanks to these Q concentrations that we estimated from the field. And then he looked at the responses of tegula in response to those more field realistic Q densities. And he sort of did the same thing that Kindle did. He looked at snails crawling out of the water and then snail grazing on algae in the experimental tanks. And we were excited to learn that even at these pretty dilute Q densities, right, you see this risk-sensitive or dosage-dependent response of tegula in response to these Q concentrations. So increased number of snails crawling out of the water and decreased grazing on the algae, even at these sort of minuscule Q concentrations that might be more like what these tegula are responding to in the field. So we can say pretty confidently that tegula can respond to these field realistic Piesaster Q concentrations in this sort of risk-sensitive manner. And so then the question becomes, given that tegula's response to Piesaster density is risk-sensitive, and there's no real functional redundancy in the system in terms of other predators that can drive these non-consumptive effects, how does variation, natural variation in Piesaster abundance in the field influence the importance and magnitude of non-consumptive effects and trait-mediated indirect interactions out in nature? So we can go back to our gradient here of Piesaster density, and like all good marine ecologists, marine intertidal ecologists, I should say, we did a caging experiment. But instead of caging predators in, we caged the prey in. So what we did is at our 4 different sites, we had these caged plots where we stocked drift kelp and realistic densities of tegula into these cages across our gradient of Piesaster density, and then simply measured tegula behavioral responses and grazing within those cages. I'll also mention that that was sort of the initial experiment, and this will come back later, but I'll describe it now. So this is a sort of a schematic example of one of our sites. So what you see are two different plots, a yellow plot and a red plot. And what we did for half of the plots is we, every 2 weeks, would stock a whole bunch of Piesaster around those plots. And in the other half, we just left it at background levels. So we'll come back, hold on to that thought. We'll come back to that later. So just looking, and here's what we did in the cages. So I actually don't even need to show you data. I could probably just show you this picture here. So here's a Piesaster crawling right up to the cage. And what you can see, if you squint, in that red circle there are a whole bunch of scared tegula crawling to the far edge of the cage away from the Piesaster. You can see them all huddled in the corner there once we took the roof of the cage off. And you can sort of see the grazing marks on the drift kelp, and you can see what we call snail polish that we marked the tegula shells to measure growth on in the experiment. So what did we find? Well, I would have been absolutely ecstatic and overjoyed if these were lab data, but these are actually field data here, right? So this is predator density across the x-axis there, and then this is cumulative and per capita grazing of the drift kelp in the cages by tegula. And what you can see, we only have the 3 points here, so not really a gradient, but you can see this nice sort of declining curve here with a reduction in tegula grazing across a natural gradient of Piesaster density in the field. So that's great. That's insightful, right, to know that under natural predator densities, natural variation in predator densities, we can get predicted variation in prey grazing responses, right? But if we want to know whether or not these non-consumptive effects and the subsequent trait-mediated indirect interactions are actually important, we kind of need to know whether they're important relative to consumptive effects of the predators in the field. And so it's actually pretty difficult to quantify consumptive effects in the absence of non-consumptive effects, because if you allow a predator to forage and consume the prey, yes, they're going to be consuming the prey, but the other prey that are around are going to be responding to that predator, right? So it's hard to disentangle those two. And it's really not known whether or not the non-consumptive effect importance is going to increase with predator density unless we can look at its importance relative to consumptive effects. So what did we do to get around this? So what we did in those same plots in another experiment is that we simulated tegula predation by just manually removing snails from those cages, right? And so we randomly assigned cage pairs within each of those plots that I described to a manual removal or an untouched treatment. And what we did, because we know that Piesaster roughly consumes, or at least can consume 25% of tegula populations in areas of overlap, we did it at a rate so that by the end of the experiment, 25% of those tegula were removed. And here's, I think, just fancy animation showing you there they go, right? Okay, so this is a little more complicated here. So we can't isolate the non-consumptive effects and the consumptive effects completely in the field, right? So the cages where tegula were removed have both consumptive effects and non-consumptive effects because those cages are in an environment where Piesaster is around. But remember, we have that gradient of Piesaster density. So what we can do is subtract off the non-consumptive effect or the trait-mediated indirect interaction from the total predator effect. So this is just an example here. This isn't real data. So we look at grazing amounts of the tegula in the cages. The non-consumptive effect grazing amount would be theoretically higher, right, than the consumptive effect amount, particularly at low Piesaster density, because if there's not a lot of Piesaster in the environment, the snails aren't going to respond very strongly. But as we remove snails from the treatment, right, there should be less grazing. So if we subtract 3 from 2, we get a negative 1, right? On the other hand, at high predator abundance, right, the non-consumptive effect and the total predator effect might not be that different, right? And so we can look at those differences across our 3 different sites that vary in piezoster density. And what we would expect to see that if there was a significant contribution of non-consumptive effects to the overall predator effect, they should be not different from 0. So the significant effect actually means that the non-consumptive effect was weaker than the total predator effect. When the confidence intervals overlap with 0, that means that the non-consumptive effect was actually as great as the consumptive effect in these cages here. And so what we see is that as you move from low predator density to high predator density, those non-consumptive, the importance of non-consumptive effects relative to consumptive effects increases. So that's pretty cool. Now, go back, if you remember, I said that part of the experiment was also that we would move, sort of artificially augment piezoster around some of the plots and leave some of the plots with just background density of piezoster. And so we can look at the results of that manipulation here on grazing, which are the top 2 panels, and then snail growth. So let's just look at the top 2 panels first. And it's cumulative biomass grazed of algae and then per capita biomass, per capita per snail, right? And the purple bars, or the purple symbols, are from the plots where we didn't artificially augment piezoster around them. And they might look pretty familiar to you because they're the same exact data from the initial plot that I showed you, right? So density-dependent response, predator density-dependent response of the prey. As natural piezoster density increase, prey grazing goes down. If you look at the blue symbols, those are the plots where we added the piezoster around the plots. And what you can see when the background density of piezoster is low, adding a bunch of piezoster near the plots actually causes a sharp decrease in tegula grazing around those plots, less so at intermediate density, and then there's no difference when the background density of piezoster is high. Right? So again, sort of showing another layer of this density dependence of the importance, or the importance of predator density on the effect, or these non-consumptive effects. The bottom 2 panels, I think, are the most exciting bits of data I've gotten since I've been at Cal Poly Humboldt. And that is because we talked about, right, if a prey is responding to a predator, it's spending less time foraging, more time out of the water, it's eating less. It should grow less. What's going on here? They're growing more. So the prey are eating less food as predator density increases, but they're growing more as predator density increases. And so that's across sort of the natural gradient here, right? So going from low to high on the purple symbols. And this is particularly for small snails. We didn't really see it for large snails, so let's focus over here. And when you artificially augment piezoster around the plots, it increases the growth of the small snails even more, despite the fact that it decreased overall grazing even more. So we were kind of mystified by this, and I thought about throwing my graduate student out of the program because I thought he was making up data. Or didn't collect the data very well. But no, I'm kidding, obviously. He did a great job, and the data are real, and we were scratching our heads trying to figure out what was going on. But a little bit of digging into ecological theory actually predicts that predators can have positive non-consumptive effects on their prey if the predator-induced reduction in prey grazing leads to an increase in resource abundance. Okay, so let's kind of walk through these 2 plots here. So in both of these, you've got resource level on the x-axis here, and then on the y, you've got both the growth rate of the prey, or sorry, of the resource, right? And this assumes, because it's a hump-shaped curve, we're assuming logistic growth, right? And then also h, which is the rate of herbivory, right? And that's noted by the linear lines, the straight lines. On the left-hand side, we have a situation where herbivory is generally low, right? And so what you can see, so the dotted straight line represents a situation where predators are absent or rare. And then the solid straight line represents a 50% reduction in prey grazing in the presence of predators. And if herbivory is low or inefficient, then really what's controlling the abundance of the resource is negative density dependence of the resource, right? And so that 50% reduction in herbivory actually leads to a lower resource level, right? when herbivory is low. On the other hand, if herbivory is high in the system, that is, the herbivores are really effective, then they're the ones that are controlling the resource population in the environment. And then a reduction of 50% herbivory by the predator can actually lead to an increase in overall resource abundance. If predators are naturally moving in and out of the system, then during times when the predators aren't there, the prey suddenly have this abundance of resource that they can consume and allocate to growth, right? And what we think is going on in our experiment in particular, so black turban snails will graze macroalgae, but what they really like to graze are diatoms and bacteria off the surface of macroalgae, particularly drift macroalgae that is breaking down. And we think that tegula is really good at controlling the abundance of those biofilms on the kelp, right? And they'll graze those all down, and then they'll start digging into the macroalgae if there's no predators around. But if there's predators around and the snails are hiding, diatoms can resettle, bacteria, which have really rapid cycling populations, can start to refill on the kelp, and then there's a bunch of food there for the snails when the predators go away. So that's sort of our working hypothesis for why we saw these positive non-consumptive effects of predators, which to our knowledge has only been shown one other time in tadpoles. So just a few take-home points from that there. So Piesaster imposed these density-dependent non-consumptive effects on tegula, and then also these trait-mediated indirect effects on algae in their environment. And the relative contribution of NCEs to CEs in the system increases as predator density increases, which is consistent with theory. And so loss of Piesaster at certain sites could lead to increased grazing by tegula, which could affect macroalgal biomass and community structure, also microbial diversity in the system potentially. And then this finding that predators can actually also positively affect prey non-consumptively. Okay, we got a little bit of time left, so I want to tell you about one more related project here. So we kind of showed that tegula can respond to these sort of really dilute cues in nature. And one of my more recent grad students, Jake Vargas, was really interested in knowing how long these cues could persist or how quickly they break down, right? That might be relevant to this predator-prey interaction. And Jake had a background working at a local brewery where he tested the beer for bacterial contamination. So he was really interested in microbes, and in particular microbial degradation of the cues. So he did a set of laboratory experiments trying to isolate the potential role of microbes in degrading the chemical cue that's being produced by Piesaster. Now keep in mind, we actually don't know what the chemical cue is that the snails are responding to, but we're assuming it's some sort of organic cue. So the first experiment that he did, he made some of our Piesaster tea, and then he froze half of the batches and left the other at ambient temperatures. And he aged both of them. So he had Q that was frozen for 72 hours, and then he had Q that sat out for 72 hours under ambient seawater conditions. And then he used this sort of now iconic crawl-out response of the tegula as a proxy for the effectiveness of the Q. And so what he found here is that under ambient conditions, which are the blue bars here with the thermostat, the Q actually breaks down pretty quickly. Tegula actually stop responding to the cue almost entirely by 72 hours. So I should just point out, so the red dotted line is the proportion of snails responding to risk cue at time 0. And then the blue dotted line is the proportion of snails responding to just raw seawater without any predator cue. And you can see that the blue bars sort of reach that control level after 72 hours. Freezing the cue actually leaves the effectiveness of the cue intact for a longer period of time, suggesting then that bacteria is playing a role at degrading these cues. So then what he did next was to physically filter out the bacteria in the next experiment to see how that would affect the degradation of the cue. But what we found was, again, something that was really difficult to explain. And so what we have here is, again, the crawl-out response of Tegula on the y-axis. Blue is the control treatments here. And then the gold bars are with bacteria present or absent after filtering them out physically. And so what we found is if you filter out the bacteria from the C star T, the snails don't respond. So that suggests that the bacteria is at least part of what the snails are responding to. So we also looked at a different way of getting rid of the bacteria from the cue. We used the antimicrobial agent tetracycline. And similar situation here. So the red dotted line is the typical Pyezoster risk cue and the time that Tegula spent out of the water in response to that. And then the raw seawater on the bottom. And then microbes absent in the purple symbols. That's the treatment that we added the tetracycline to. And then we had a procedural control where we added stuff to the cue water that wasn't going to affect the microbes. And what you should focus on is time 0 here. Again, what we see is that if the microbes aren't in the cue water, the Tegula respond half to about 1/3 as much if they did when the microbes were present. So what this suggests is that microbes are actually playing an important role in Tegula recognizing and responding to this Pyezoster chemical cue. Now, notice that as we move on in time, both sets of snails stop responding to the cue. So microbes and bacteria probably still play an important role in degrading the cue over time, but there's at least some subset of the bacterial community that might actually be activating whatever chemical it is that Tegula is responding to. So we're pretty excited about that. So what we think is microbes are probably some key constituent to Pyezoster risk cues where they play an important role of activating the cue so that Tegula can respond to it. But they also play a role in degrading the cue as well. And so important now to maybe think about how the microbial environment associated with sea stars or any marine predator might actually influence these chemical signaling mediated predator-prey interactions. All right, I just want to say thank you to the funding sources, my collaborators, and definitely want to give a big shout out. I mentioned 3 students here, but there's a whole host of other undergraduates in my lab that helped out with these projects, and other graduate students that dedicated their time to helping out their fellow graduate students on these projects as well, too. So thanks so much for being an attentive audience and for coming to the talk. I appreciate it.
Any questions for Dr. Bourdeau?
In the back first, and then Sarah.
Okay, back first. Bruno first.
Hi, thanks for the talk. It was super interesting.
Yeah.
I was wondering if you were aware of maybe any like microbial communities that are specifically associated with like the surface of sea stars, maybe that are known to live on their skin per se, and whether or not any of those were maybe identified in the sea star juice compared to some that might just be floating in the ocean?
So right at the same conference that Jake presented these data, there was a postdoc, I forget where she was, I think at UBC, who was looking at microbial communities of West Coast sea stars. And we talked with her a bit, and we actually think that we haven't sort of looked at what the bacteria associated with our Piesaster are. I think Jake plans on doing that for his dissertation work, but we think it's probably gut bacteria in the Piesaster that are important in this case here. That's what we're hypothesizing anyway, yeah. But we don't know which ones.
Well, still exciting. Thank you.
Yep.
Great talk, thank you. My question was kind of more about the abiotic factors that might be playing a part. So I was wondering, just in all these projects that have been going on, has anyone been considering the effects of abiotic factors? Like the biggest ones that I'm thinking of are like disturbances, so like from storms mostly, and then temperature as well. I would imagine that would affect how those chemical cues are moving.
Yeah, it's really interesting that you bring that up. I actually had very limited hope that any of the field experiment was going to work because, as you know, right out on the Humboldt coast, it's pretty wave exposed, and for the most part, the sites that we chose are moderately wave exposed sites. So there's quite a bit of wave action on that part of the coast. There's a lot of river outputs, so there's a lot of sedimentation, and you might imagine that in a real flowy or hydrodynamic environment, Q is going to get washed away pretty quickly. It's not going to accumulate, and the prey might not be able to respond. So we're actually kind of surprised at how strong a response we got. But certainly hydrodynamic forces would sort of act against the Q being transmitted from the predator to the prey effectively. Temperature is an interesting one too, especially if the bacteria play a role in activating the Q, because increases in temperature, you might imagine, could increase the life cycle of the bacteria, maybe increase their metabolic activity and make the Q more effective. I mean, who knows? But yeah, certainly lots to be done there in terms of looking at how abiotic environment affects the signaling.
It's a great talk. Thank you very much. So I was wondering about the consumptive effect you were modeling there, and that so you modeled it by removing snails from the environment, which impacts the population, but I don't think snails. I don't know enough about snails to know if they can count and recognize that their friends are gone, but it doesn't seem like they can.
You'd be surprised.
But so I was wondering, is there maybe an effect of like when a Piesaster eats a snail, it crushes the snail, and presumably there's some scent and stuff released into the environment from that. Is that incorporated into any of this?
Yep, so that's a great question too. And we know from a lot of predator-prey chemical signaling studies that if a predator is consuming conspecifics of the focal prey, then the prey response is actually going to be stronger than just the response to the predator itself. We've actually done those experiments with tegula. So Piesaster sucks tegula out of the aperture of the shell and it externally digests it. And at least as far as our experiments have shown, the response to Piesaster itself is actually stronger than the response to just damaged tegula in this case here. And the way we damaged tegula was probably not great because we actually did macerate them, which is not how the sea star consumes them. But we think the Piesaster Q itself, or the microbes, whatever is behind it, is what's driving this. I should note too that we did control, so the Piesaster that we used in all of these studies were starved for several days to weeks prior, so that we sort of factored out any effect of their diet prior to the experiment.
Interesting, thank you.
Mm-hmm.
It's a very interesting talk. So I have two questions actually. Sorry. So you showed us this graph where the bacteria was filtered out, and then that was like the 0 times 0, and then 0.5, and so I think it's just the slide of 4.
This one.
No. So the times 0. Yeah.
This one.
Yeah. So I'm just curious as to why. So you said to focus on like the time 0 where it kind of decreased by about 75%-ish. Why did it increase in the next half an hour, 1 hour or so, before it drops again?
So that gets at the very reason why I wanted you to focus on the 0, is because we don't know.
Right. So is the 0 just right after you filter it, and then you put those.
We give them in our experiments typically a 5 to 10 minute acclimation period after the Q is added. So time 0 is really about 10 minutes in, and then this is time after time 0. So
Right.
we actually don't know why. So we have some hypotheses. One is that maybe the tetracycline was good at killing off some of the bacteria in there, but not all, and then maybe it killed off, maybe there's some trade-off between resistance to tetracycline and ability to activate the Q, and so it killed off the ones that activated the Q but left the others. We don't know, and I don't know enough about microbial life cycles and activity or the microbial diversity in our system to kind of know what went on here.
Yeah.
Could also be,
There's a lower concentration of bacteria, just took them longer to synthesize.
could
Yeah,
have taken
that's-
them long. Yep.
Yeah, that's what I was thinking.
Yeah.
Also, another question. You had like 4 sites in the at the ocean.
Yes.
Initially, so 4 natural sites.
Yes.
And then there were 3
And then
data
there were 3.
points. So like something happened.
No, so what happened, so we were doing this kind of right after the sea star wasting outbreak, and
Right.
one of our sites was basically a 0 Pisaster site. And CDFW did not want us to move Pisaster from our other sites to that site to create our experimental conditions. So we didn't use that 4th site because of that.
Cool. Thank you.
Yep.
Just one hypothesis before I pass to Madison on the tetracycline.
Yeah.
I don't know if that antibiotic affects primary cilia, but I know other ones might. So maybe the more exposure to it over time in that water could have affected the snail's ability to sense or something to stress them and make them run out of the water. Potential.
We did do some pilots just adding tetracycline to raw seawater with the tegula to see if they responded differently just to the addition of, didn't seem to, but shorter term stuff, so who knows, yeah.
I was really interested in the positive response of the tegula. Do you know of any other like intertidal species that shares that same resource of like the stuff on the dried kelp, and would they also have a positive response because the tegula?
Yeah, I mean, I guess if you have a resource that can grow quickly and turn over quickly, and then an herbivore or a consumer of that resource that is an effective consumer of that resource, then that theory should apply to any situation where those two things happen, right? Like a rapidly cycling resource and a really effective consumer of that resource.
Did you still have a question?
No, I was similar to something. I was going to ask about the interconspecific heat detection.
Okay. Any other? Oh, I'm sorry.
No, it's okay. It was more methods based anyways.
Yeah.
It was about that one student project where they diluted the predator Q.
Yeah.
I was curious, like, since we don't know what the predator Q is, how did y'all determine like how many dilution steps to use, and like how do you know it's like really reflective?
So we don't. We did our best estimate. So what we used as our sort of Q is just an individual Pisaster. So we had from the field, aerial densities of Pisaster, right? And then we just decided, well, okay, the Pisaster is living basically in this column of water, right? So we can convert the aerial density to a volumetric density. So 1 Pisaster per cubic liter or whatever, right? And then, so we used our estimates of that from the field to, we had our Pisaster incubated in a bucket or a tank. We knew the volume of that tank, and so we just used that to then calculate how many dilutions we had to do to get to that field concentration.
So it's almost like, experiencing the snail right next to it versus like a few meters away or whatever.
Yep.
That was a great talk, by the way. Thanks so much.
Oh, thanks.
Really fun, yeah.
Cool. Any more questions in the room? Not sure if we should check Zoom. See if
Oh.
there's anyone on the chat. On there. Hi. Oh, hi. I can't see. No question. Okay. No questions on Zoom.
Okay.
Could I ask one more then?
Of course.
Sorry. I had one and I wrote it. Okay.
Yeah?
And this This is like when someone says, I know how to juggle, and someone says, great, how many? Do you know how to do n+1? Because this is a ton already. But because you mentioned cages,
Yes.
so I'm like, the other common intertidal thing to do is transplants. And so I'm wondering whether tegula from low-risk sites would have different responses when introduced to predators than tegulas that are accustomed at a higher risk site? And if you've explored that or have any hypotheses.
Yeah, so I had a student do that very experiment, and it turns out that at least across the 4 sites that we have, which goes from 0 to a lot of Pizoster, all the tegula respond similarly. Now, it's very likely that prior to C-star wasting, that gradient wasn't as stark as it is now, and so the tegula that we tested from our 0 site probably have an evolutionary history with, historically with more abundant Pizoster there. So we really probably need to do it over a larger,
Cool.
both
I love
time
that you did
scale
that.
and spatial scale.
Thank you.
Yeah.
All right, well, thank you.